Good evening student, welcome back. In this video, we will try to learn about the unit membrane model proposed by Robertson. Just to revise, we have already seen a sandwich model of plasma membrane which is proposed by Danieli and Davison. Okay, and now we are learning about a second model, that is unit membrane model of plasma membrane which is proposed by Robertson. A unit membrane model proposed by Robertson is actually a upgraded version of sandwich model. The basic information of a unit membrane model will be same just like sandwich model. According to this model, the plasma membrane is also a trilamellar structure. This plasma membrane is consist of lipid bilayer, where the lipids are phospholipids, and the actual arrangement of these lipids and proteins is PLLP. So there will be a lipid bilayer which is sandwiched between the layer of proteins. But there are two additional things which is mentioned by Robertson. Number one, he has mentioned the proteins in the plasma membrane are actually beta fibrous proteins, which is a difference compared to a sandwich model, because according to the sandwich model, the proteins which are present in plasma membrane was alpha globulin proteins. Now according to Robertson, there are beta types of proteins which are present in the plasma membrane. And secondly, the Robertson has mentioned the dimensions of plasma membrane. According to Robertson, the dimension of plasma membrane is 75 Armstrong, in which he has proposed there will be a 20 Armstrong layer of protein, plus there will be a 35 Armstrong layer of lipid bilayer, plus there again a 20 Armstrong layer of protein. So according to the Robertson unit membrane model, the dimension of plasma membrane is 75 Armstrong. This is the additional point which he have given. But again, the model has failed to prove certain properties of plasma membrane. Let's check out. If according to Robertson, the lipid bilayer is arranged in which the globular heads are facing outside and the fatty acid tails are facing each other, and the protein layers which are present here, the beta type of proteins, makes the layer non-dynamic, because this type of structure also do not proves, number one, limitation of unit membrane model. Number one, it cannot prove selective permeability, because whenever a substance, or say let's say water soluble substance, will try to enter the plasma membrane, then it can go up to this level. As this much part of plasma membrane is hydrophobic, so a water soluble substance cannot pass through this membrane, so it do not help in proving the selective permeability of membrane. So this is the first limitation of this model. Then secondly, it cannot prove the variable ratio of proteins and lipids. See, take a section of this plasma membrane. If we take a section of plasma membrane, then here 1, 2, 3, 4, there are 4 molecules of lipid. So lipid we are having So there are 4 molecules of lipid, and here 1 2 3 4 there are 4 molecules of proteins. Clear? So if you want to change a ratio, then the structure will change, and if you do not change the ratio, that means the membrane is rigid. So this model also failed to explain the fluidity, that is dynamic nature of plasma membrane. And these are the 3 reasons due to which these 2 models got failed. And basically they got failed because at that time they was not knowing the actual structure of plasma membrane due to limitation of electron microscope. इलेक्ट्रॉन माइक्रोस्कोप ही नहीं था और इसलिए उनको प्लाज्मा मेम्ब्रेन का एग्जैक्ट स्ट्रक्चर ही पता नहीं चला. व्हाट दे हैज प्रपोज्ड आर द हाइपोथेसिस. Okay, just to summarize everything, we have already seen 2 models for the structure of plasma membrane. पहला वाला मॉडल हमने सैंडविच मॉडल देखा जो डैनियल एंड डेवसन ने प्रपोज किया था, और दूसरा वाला मॉडल हमने देखा यूनिट मेम्ब्रेन मॉडल जो रॉबर्टसन ने प्रपोज किया था. दोनों ही मॉडल में जो लिपिड और प्रोटीन्स थे उनकी अरेंजमेंट सेम थी, यानी लिपिड की बाइलेयर प्रोटीन के लेयर्स के बीच में सैंडविच की तरह प्रेजेंट थी. लेकिन ये दोनों मॉडल इलेक्ट्रॉन माइक्रोस्कोप के इन्वेंशन के पहले आए थे, इसीलिए ये दोनों मॉडल फेल हो गए इन टेलिंग कि सिलेक्टिव परमेबल नेचर प्लाज्मा मेम्ब्रेन का किस वजह से आया. दूसरी चीज, ये मॉडल्स ये नहीं बता पाए कि प्रोटीन और लिपिड का जो वेरिएबल रेशियो है वो कैसे आया. और तीसरी चीज, वो प्लाज्मा मेम्ब्रेन की फ्लूइडिटी यानी डायनामिसिटी यानी हिलने की ताकत, इसके बारे में वो हमें बता नहीं पाए. इसीलिए ये दोनों मॉडल फेल हो गए. और आफ्टर द इन्वेंशन ऑफ इलेक्ट्रॉन माइक्रोस्कोप यानी इलेक्ट्रॉन माइक्रोस्कोप बनने के बाद सिंगर एंड निकोलसन ने एक बहुत बढ़िया मॉडल हमारे सामने पेश किया प्लाज्मा मेम्ब्रेन का, जो वर्ल्डली एक्सेप्टेड मॉडल है, एंड दैट पर्टिकुलर मॉडल इज कॉल्ड एज फ्लूइड मोजाइक मॉडल. So in next lecture we will try to learn about the fluid mosaic model proposed by Singer and Nicholson. Thank you.